Thanks for the great turnout. I'll pretend it's for me and not the pizza. This will be the first chats that I've done, but I've certainly enjoyed attending almost all the other ones. So as those of you who have been to them before know, it's meant to show you an array of the research going on campus and in the department. And so I'll be presenting a bit about what my research group does, not in its entirety, but a few elements therein, but also in the context of the broader research enterprise, and particularly as applied to protein engineering. Right? So I'll focus on discovery and evolution of both diagnostics and therapeutics for clinical use. So proteins are very, very powerful for a variety of different applications. And so, for example, they can be used as catalysts when they're enzymes. And so this is superoxide dismutase. It has the ability to turn over 1 million reactions per second, which is very important for all of us because what it does is clear off oxide radicals inside of our cells so they don't cause too much damage. And so it needs to be a very fast, very potent enzyme to do that. They can also be binding agents or recognition agents, such as the antibodies. So the native immune system has the ability to identify any non-native, almost any non-native substance, and then elicit an immune response to clear that from the body with very, very high functional efficacy. Not perfect, but very, very effective at that. Then they can also be structural or movement-based agents in terms of proteins. And so here's a video of microtubules and a kinesin motor walking along that microtubule, carrying cargo throughout the cell. And this movie, if you will, is actually slowed down quite a bit. They can traverse over the course of a cell in a matter of a few seconds. So very, very efficient at what they do. Right? From a structural perspective, they're able to achieve this array of different actions and functions despite fairly simple structure at the small level, but then at the same time very complex structure at the three-dimensional level. So at the chemical simplicity level, it's simply a polymer of amino acid monomers built here. But if you then consider that this side group, which I'll just call R, can be any one of these 20 natural amino acid chemistries, that's a lot of chemical diversity that is then available. And then when you string that together in a long polymer of tens or usually hundreds or even thousands of these amino acid monomers, you can have a tremendous array of complexity and diversity in terms of the structures, and therefore the functions, such as the ones I highlighted on the last slide. Right? So key question then in protein engineering is, can we harness this diverse molecular power to create novel function, right? So these are examples of native function. Can we modify those functions or even find brand new functions, right? And so that in general is the field of protein engineering. And so can we put the appropriate amino acids in the appropriate locations to drive new or improved function? And so I'll share a bit about a few examples from my lab in which we engineer proteins to detect cancer, combat antibiotic-resistant infection, as well as catalyze substrate degradation for a biomarker-based approach. Okay. So what is the protein engineering challenge? The challenge is we have some function in mind that we'd like to achieve, either new or improved function thereof, but we need to figure out what structure and even more basically what sequence will achieve that particular function. And we can approach that two different ways. Well, ultimately what I'll say is that the combination of these two approaches is actually what's most effective. So one approach would be rational design. In theory, we know what the sequence is, therefore we should be able to use physical principles to predict that structure and therefore predict that function of what it looks like. Unfortunately, that's a challenging thing to do for multiple reasons. The two dominant reasons would be that there's an immense conformational space available. If I have a particular protein sequence at the amino acid level, the number of possible structures that it theoretically could fold into is immense. And so calculating all those possible conformations and figuring out which one is the most stable is very, very challenging. On top of that, we have, I'll say, imperfect energy calculations to be able to model molecules of this size, of thousands of molecular weight. And so the sequence goes to structure goes to function problem is tractable but not perfectly solvable. And that makes pure rational design challenging to do, yet there are definitely advances in that space. On the other end of the spectrum, we could go to experiments and find out which molecules are indeed the best. Unfortunately, sequence space, as I'll call it, which is all the possible protein sequences we might have, has three elements which are challenging to the protein engineering problem. The space is immense, so there's 20 monomers and therefore 20 to the nth power of possible proteins of length n. The space is very barren, which is to say most of these sequences do absolutely nothing of interest, and particularly not what we want them to do functionally. And the space is very rugged, which is to say you can be very close to a functional region of sequence space and yet not have that function. Or you can have some decent level of function, and make one mutation and fall off the fitness cliff, so to speak, in terms of losing that function altogether. So this combination of immense, barren, and rugged makes random searches of sequence space very challenging to do. So even with very high throughput techniques, it's very easy to find no function in that space as well. Yet there's very, very good progress made in this space as well. But ultimately, it's the fusion of those two things, which I'm going to explain today, is the most powerful. But first, I'll just highlight each of those one individual things in slightly more detail. So on the combinatorial side, the idea is, can we make a whole bunch of different protein variants and then test them very efficiently for that function or set of functions of interest and find the ones that work well, right? If we're efficient enough in that search, this should and can work. And so we take the 20 natural chemistries, or some subset thereof. We put them into our protein of interest, but we do this at the genetic level. And so we can very efficiently synthesize many, many genes, millions, billions of genes very, very cheaply and easily now. We can then take those genes, put them into a cellular host, and have the cellular host make the protein for us. But then we still need some assay or some experiment where we can test them to see how functional they actually are. So I show one example here of what's called e-surface display, but there's many other applications used as well, and I'll detail this one in a little bit. And then we can couple that to some additional tests for different functions, such as binding function shown here. And then, in some cases, that's it. All it really is discovery. We make a whole bunch of different candidate proteins. We efficiently evaluate which ones are functional. We pull out the most functional, and we're done. We've discovered our very functional protein, and we stop. But in many cases, because of the immense size, the barrenness, and the ruggedness of sequence space, that doesn't work very well. We have a protein that's minorly functional, but not nearly as functional as we need it to be. And so we then use directed evolution. We pull the DNA encoding for the good protein out of the cell, modify it in some way to create a new library that's better than our initial naive library. This one is now informed by what was functional before, and we iterate this process. And so this process of directed evolution recently won the Nobel Prize in partnership with phage display, which is an efficient way of doing discovery as well. So on the other end of the spectrum, there's the rational approach. In theory, we should be able to systematically evaluate protein candidates computationally and or theoretically and predict which ones are going to be functional. And again, this can be done, although there's still a long way to go to make this as robust as we would like it to be. And again, that comes back to this sequence structure function problem. Now we basically want to do it in reverse. I have a function of interest. Can I pick a structure that will achieve that function? And therefore, can I find a sequence that will give us that structure? Right? So I put down here one example that has worked very, very nicely. This is out of the lab of David Baker, as well as some collaborators on that, where they've taken a structural analysis approach, considering both inter and intramolecular interactions, to ultimately design a binding ligand. So their aim here was to make a protein that binds to hemagglutinin from influenza. In particular, they wanted to make a molecule that binds to this particular location or epitope on the protein. So what they did computationally, using a piece of software called Rosetta that they designed, was to determine where could I place amino acids in a protein that would interact favorably with this particular protein target. And they found that if they put these three particular amino acids in those precise conformations, they would prefer to be in the bound state versus in the unbound state. And so if they then built a protein that had those amino acids in those conformations, that protein would potentially then prefer to be in the bound state rather than the unbound state. And so computation, they sampled many, many different protein topologies, many more than the ones just summarized here, to find particular protein shapes or topologies that would have these three particular amino acids very close to that exact conformation. And then they looked at that in more detail and said, if we build that particular protein, what will the rest of the amino acids in that protein do in terms of interaction as well, both intramolecularly and intermolecularly with the binding target? And then they iterated that process to improve the design and made basically computational mutations to the protein to evolve it to be better, all in silico for starters. Then they went into the lab and actually built the proteins, or even a list of candidate proteins, and tested which ones worked well. Out of numerous candidates, many of them did not work, but there were a couple that did work reasonably well. And they then solved the crystal structure, co-crystal structure of that newly designed protein in complex with the intended hemagglutinin target, and found that it had a very similar conformation to what they designed it to do. And importantly, experimentally, it bound to its target with modest affinity, but nevertheless a fully de novo design of a protein binder. They then followed that up with more experimental directed evolution, akin to the last slide, to make it bind even more strongly and selectively. So that is certainly one viable approach, nevertheless a challenging one to do. Another approach from the rational perspective is genomic analysis. If we're trying to make changes to a protein, but we're concerned about the ruggedness of sequence space and we're going to make a mutation that's going to ruin the functionality of my protein, could I look at nature and say, has nature ever had a similar protein, and how well did it tolerate the mutation that I'm about to make to it? So I can go to now the growing database of sequences from different genome sequencing efforts and look, are there similar proteins in nature? And if so, what mutations do they tolerate or what mutations do they not tolerate? And that can guide us towards preferential mutations that will help instead of hurt. But really, as I've said a few times now, the power is at the fusion of these two things. The power is at the idea of putting these ideas, the structural approach and the empirical approach of Rosetta and that computational framework, the genomic approach of looking at what nature has already tested for us, as well as a few other things I'll mention in the coming minutes, to predict which library, a collection of protein variants, is more likely to work. So rather than using design to design one, we're going to use design to design many different protein candidates, and then go through the high throughput approach of finding which ones are most efficient via the strategy outlined before. So now let's jump into a few particular examples. Can we put this to work in the lab and on the computer to make proteins that have particular applications in mind? So for the first case example, can we make a protein that can selectively detect cancer and give us molecular information about that? Why is that important? Well, I don't think I need to motivate the importance of cancer and its detriment to health and society. I'll just leave it at that. But I'll really highlight the need technologically to have better, earlier detection, as well as more molecular information about the patient's disease. On the early detection side, in many, many cases, earlier detection improves patient outcomes dramatically. So this is just a snapshot from a few years back now. Nice schematic from Wired magazine showing across several different cancer types. And certainly all cancers are different. Not all ovarian cancers are the same. Not all lung cancers are the same. But nevertheless, in this case, grouping them together for the purpose of this schematic and showing the 5-year survival for the patients as a function of the stage of detection of their disease. So there's the within the spectrum of the unfortunateness of cancer, the better case scenario of prostate cancer, where by and large it tends to be, if detected early, and in most cases it is detected early, this thickness of this line, the survival, 5-year survival, is actually quite high. Unfortunately, for the half or so of the patients for which it's detected late at stage 4, when it's already metastasized or spread throughout the body, the 5-year survival is quite a bit lower. On that spectrum, on the much worse side of the spectrum, pancreatic cancer, for example, unless it's detected early, the odds of 5-year survival are unfortunately very, very dim. And based on the thinness of this line, unfortunately what that means is very few patients actually have early detected pancreatic cancer. The grand majority are detected even after metastases has already happened, and therefore survival is unfortunately very, very low. And so if we had technology for earlier detection, for example, of pancreatic cancer, that would be very beneficial to patients. On top of that, molecular information would be very advantageous as well. In many of these cancers, there are good therapeutic options, but we're not getting them to the right patients at the right time. And so many of these molecularly targeted therapies work very well on a subset of that patient population, but we don't have a diagnostic to stratify which patients respond versus which patients don't respond. So if we could characterize which are which, the physicians could give the right treatment to the right patient and come up with a different strategy for the patient who's not going to be responsive to that particular molecularly targeted therapy. I just put one example here from a trial targeting a particular cancer biomarker called MET. And so Genentech, now Roche, engineered an antibody that targets this particular receptor and antagonizes its action in lung cancer. And went through a lot of very clever, very rigorous protein engineering to make this molecule act in the way it was supposed to act, and to antagonize or block the receptor's activity without causing additional signaling. And then ultimately went into the clinic to test how well that worked. Unfortunately, the clinical trial was grouped with all the patients together receiving this therapy on top of the current gold standard here. So the current gold standard is shown here in yellow, and the patients that were put into the half of the trial for which they got this new engineered antibody are shown here in blue. But then after the fact, additional analysis was done and published in this paper to show which patients actually had very low levels of this biomarker MET and which patients had very high levels of the biomarker MET. For the patients whose tumors were being driven by this particular biomarker MET, that's what I'm calling MET high here, the addition of the antibody improved their long-term survival dramatically, right? Still not what we want it to be. Everyone survives. That's ultimately the goal. It's still dramatically improved relative to the current clinical standard. So that's great. Unfortunately, the patients that happened to have a low level of MET expression not only did they not get a benefit because their tumor wasn't being driven seemingly by MET, but they actually saw a slight detriment. And so when all of these patients were grouped together into the same trial, ultimately it was deemed that the molecule was not good enough and safe enough to use, and so it is not in the US FDA approved for use. But seemingly, this is a retrospective analysis, but seemingly it suggests that had there been a diagnostic that could stratify the groups from each other ahead of time, then this would have been very advantageous for those particular patients. And so, in short, early detection and better molecular information can be very powerful. But to do that, we need some technology to achieve that. And so one approach to that, of many options, one approach to that is molecular imaging. And so the idea here is, can we make a molecule that selectively will target that biomarker and provide information about its activity or its location or its concentration? And this can be done, as I alluded to, for patient stratification. So this approach is showing taking an antibody as the engineered protein, labeling that with a positron-emitting radioisotope, administered it into the patient's bloodstream, waiting a while for that molecule to distribute and clear when not bound to the target of interest, and then taking a PET scan to see where's the location of that molecule throughout the body. And so you can see in particular locations, unfortunately, the metastatic spread of these patients' tumors. But not only where it is, but the fact that in this case it's a HER2 targeted agent, that these tumors are being driven by that. There's also value in early detection and utility there. There we probably would not want to use PET scanning because of the cost and the ionizing radiation associated. But ultrasound, for example, would be a very compelling option. Relatively cheap, very, very safe, ubiquitous throughout the world. And so this is an approach where we can take a bubble, label it with a targeting protein that binds selectively to a biomarker, and perform ultrasound imaging on those patients. You can also use it after detection and stratification to monitor how that patient is doing. So in this case, it's taking a glucose analog. You take glucose and you take off one hydroxyl and replace it with an F18 molecule, and then you can image glucose metabolism. In addition, outside of clinical use, it's also useful for scientific study as well. But to make all of these different approaches of molecular imaging actually used, we need engineered molecules that selectively bind to the markers of interest. We need either antibodies or small ligands or what have you that can serve as those molecular targeting agents. So that brings us back then to the aim of protein engineering. How can I efficiently engineer ligands to serve as the delivery vehicles for these different diagnostic moieties? Okay. And so that comes to the question then of, to make this work efficiently at that hybrid of rational and combinatorial engineering, how do I efficiently find this ligand? And so for this particular case, we're going to use this particular protein called the affibody. It's a 58 amino acid protein in size. And the question then is, how can I use a rationally guided approach to pick which amino acid options I want at particular locations in this affibody to efficiently evolve it to bind to a biomarker of interest? Right? So in this case, we took a guide from nature. Well, how does nature solve this problem? The natural immune system in almost all organisms, including us, is this antibody molecule. And it has binding activity at the two surfaces shown here. But it doesn't take the naive approach of saying, I'm just going to put the 20 natural chemistries anywhere I want in that protein, or equally in that protein. I'm going to put them in a particular location, and I'm going to put a higher level of some amino acids relative to other amino acids, particularly high levels of these 3 amino acid side chains. But it's not just high levels of these throughout the antibody, it's different levels of each amino acid at each position within that protein in a very particular way that has evolved over time. But that's how nature has already evolved it very functionally. Can we do that in the lab synthetically to achieve an efficient approach in our molecule of interest, which has some own benefits relative to the antibody? Right? So the question is, what should our distribution look like for our particular molecule of interest? And so that's where we went to the rational perspective and said, how can we ahead of time predict what might be better? So we used a whole slew of different techniques. We looked at some earlier work from us and others in the community that had tried to evolve this particular affibody molecule to make it bind to various different biomarkers. And we looked at which amino acids are preferred at which sites in that protein. We also computed the stability or instability of making different mutations at different sites. We also looked at different proteins that look very similar to this, i.e. the natural sequencing approach. What mutations has nature made to this protein successfully? We also asked, chemically speaking, what amino acids are better at binding to targets than others. And lastly, because this is a helical protein, we asked the question, which amino acids favor alpha helical secondary structure. And we put all of that together to make a predicted effective library, but then ultimately went into the lab to test how well did this actually work. Okay? And so what we did is we used this technique that I alluded to before called yeast surface display. We put a library of different genes, all using these principles as guiding designs in terms of which genes we should make in terms of particular amino acids at particular positions. And we put those genes, about 1 billion different genes encoding for proteins similar to this one. And we put them into baker's yeast, Saccharomyces cerevisiae yeast, very similar to the yeast that you'd bake with just a few genetic modifications. One of them is to express higher levels of this mating protein called agglutinin-1, or AGA1. Okay? So when we put the gene in encoding for our library of different alpha bodies, we put it in as a genetic fusion to the mating partner protein AGA2. Right? And so what happens is it makes our protein of interest, sends it outside of the yeast cell, but those two mating proteins interact with each other and form 2 disulfide covalent bonds. And so we now have our protein of interest on the surface of our yeast cell. And so we can interrogate its activity, in this case binding to our cancer biomarker, in the context of this. But we can do that all in one test tube, because inside of that yeast cell is the gene encoding for that particular protein of interest. So we put all the proteins together, we expose them to the biomarker, and the molecules that happen to be good protein mutants bind to that particular target and are now either magnetically or fluorescently labeled. And we can pull those out using a technique called flow cytometry. So we can evaluate one by one each one of these cells. So every dot is a particular yeast cell that has a particular protein on its surface. And we can evaluate how much binding do we get to that target because the target is fluorescently labeled. We also normalize for how many alpha bodies on each yeast cell by using a second binding protein that gives us a signal there. So all the molecules, all the yeast that are in this polygon here are molecules that express a full alpha body on their surface and bind very nicely to the particular biomarker of interest. And so we collected all of those. And we did that for several different targets to try to learn how effective was this library design strategy before we ultimately applied it to the particular cancer biomarker problem. So what we did then is took all of those tens of thousands of binding proteins and we sequenced the genes that encoded for them. And then we evaluated site by site throughout that protein which amino acids do they prefer to be. So red means it prefers to be that amino acid. Blue means it prefers not to be that particular amino acid. And so many of the sites are kind of neutral, so to speak, but many of them have strong preferences in one direction or the other. And so if we take this information and feed it back into that design loop, we can make a second generation library, which is now much more efficient at evolving binding to the function of interest. And so we can compare this designed second generation library to an uninformed or naive library, and we get a lot more binders in the design versus the naive approach. In addition to that, the designed library uses molecules that are much more stable than the naive library approach. And that's actually likely not coincidental, but rather a driving reason for why it's so evolvable. Right? So then we put this to work on making those actual cancer biomarker targeting agents. So in this case, the biomarker of interest is called B7H3, also known as CD276, these kind of cryptic immunological names that we have. This was chosen because our collaborators at Stanford University were evaluating particular biomarkers that are present in tumors and in particular tumor vasculature, because they have this very clever technology called molecular ultrasound, where you take a couple micron size perfluorobutane core, liposome, and then you label that with binding ligands. And what this does is provide very good contrast on ultrasound imaging, because we don't have a bunch of perfluorobutane gas inside of us. And that gas provides a nice acoustic contrast. And so you can engineer this entity, administer it into the patient, or for now in mouse models of cancer with this. And what you'd expect to have is these molecules floating throughout the bloodstream. But in the presence of the biomarker, many of them will now be bound to those particular cells and tissues. And so you'll have a higher concentration there. To differentiate which bubbles are bound to actual target versus which ones are just flowing through, they provide a high energy, very brief, high energy pulse to destroy all the bubbles in one region. Then a moment later, new bubbles that were not in that region will be flowing through the bloodstream and now occupy that region. So you can compare the before to the after. And so the after will be the local density of just the blood flow, but the before will be the local density of blood flow plus all the bond molecules. And so that differential between those two imaging time points will tell you how much biomarker you had present. Right? And so to make this work, it needs to be a vascular target, a target that's open to the bloodstream for our bubbles to bind to. And so they did a lot of work looking at clinical samples to evaluate which biomarkers would be indicative of disease. And this B7H3 molecule emerged from that. And this is just tissue staining, validating that this is indeed a molecule that's much more present in cancer vasculature than it is in healthy vasculature. So the aim now is to engineer a ligand that can do the role of making sure it binds to those targets of interest. And so we took Danny's improved Affibody-based library, Danny, the graduate student who led that particular project. So we took that improved library that has site-wise design diversity, we put it into our yeast display system, and we sorted it for binders to B7H3. In this particular case, to make sure that we had binders to genuine B7H3, we not only used it as a fluorescent target via that cytometry strategy I just described before, we also exposed it to full cells or fragments of cells that also express this biomarker to make sure we were presenting genuine B7H3 and not just a recombinant mimic thereof. And that's work that Larry Stern and Patrick Laun, as well as Ali Kobe in the lab, have led. And they found that to make this cell-cell interactions, we have yeast cells on top of cancerous cells in a petri dish. To make sure that that interaction goes well, we found that we actually had to lengthen this linker to get our Affibodies farther away from the cell. So considering the geometric constraints at hand, we found that a longer linker was much more functional at finding those agents. So putting these different technologies together, improved library design plus improved cell-cell interaction, led to a functional molecule. And so we took that functional molecule, we put it onto this perfluorobutane cord system, put it into a mouse model of cancer, as well as healthy mice, and did molecular ultrasound imaging in collaboration with our colleagues at Stanford, as I mentioned before. And we can see a very nice differential between the cancerous tissue and the healthy tissue. And so now this is continuing to be optimized to ultimately, hopefully, translate into the clinic. Okay. So let's move on to the next application then. So can we engineer proteins to not just be diagnostics, but to be therapeutics themselves? And in particular, can they combat antibiotic-resistant infection? Right? So antibiotic-resistant infection is a critical and growing challenge. Right? So resistance continues to increase. And so this is VRE and MRSA, two very problematic infections in the clinic. And this data is old, but the trend is very clear that the frequency of resistance continues to grow and grow. And it's also a very growing of importance. It's estimated that this will yield upwards of 10 million deaths in the US by the year 2050, rivaling that of cardiovascular disease and cancer. In addition, innovation is waning. The amount of antibiotic discovery is dwindling with time, and the speed of those molecules being resisted against is unfortunately quite quick. And so our proposed solution is to use antimicrobial proteins and peptides, or AMPs, as a solution. So there are many different AMPs and many different modes of action that they have, which is one benefit of them. In addition, they tend to be more selective than the broad spectrum antibiotics that we have in the small molecule realm. So we can use these unique modes of action, selective modes of action. They can also be very efficiently engineered. So if we do develop a new AMP and use it, and eventually the microbes develop resistance, which is likely to occur, we can go back to our protein engineering platform and efficiently find the next version, and the next version, and the next version. So I think kind of a sustainable engineering approach is actually quite relevant and important here. In addition, these molecules can be engineered to be very potent against a particular pathogen of interest. And the real a primary aim here is this combination of not only potency, but also that selectivity, so that we're only hitting the pathogen and not wiping out the rest of the healthy microflora, which is very valuable for many different functions inside the body. And so we want to be very, very selective. And we also need that efficiency of engineering for when resistance ultimately does reemerge. Right? And so this is one particular example from a different group showing a difference between a particular small molecule and antimicrobial in terms of the types, the diversity of the microflora after administration of this molecule. And so after administration of the small molecule antibiotic, there's a dramatic change in the particular distribution of the microflora, which is generally speaking not desirable. Conversely, after the antimicrobial is delivered, yes, there's a slight change, but very, very slight relative to the dramatic change shown on top. Now, this is one particular case example, and we need to look at what this looks like for many other molecules as well. It's also important, as I've highlighted, that these can be efficiently engineered, either using the yeast display system I talked about before, or other strategies that we and others are working on, such as co-encapsulation of both the AMP-expressing probiotic host and the pathogen inside of these agarose droplets, that we can then interrogate how active is that particular AMP relative to others. So a particular example here, some work that Seth Ritter and others in the lab have been working on. So C. perfringens is a particular pathogen which is a substantial healthcare problem. It's spore-forming gram-positive bacterium. It yields upwards of about 1 million or so foodborne illnesses every year, and therefore we'd like a solution to combat that. So one possible approach would be to take endolysins, which are catalytic enzymes that chew up the cell wall. So there's a pair of domains, one domain that binds to the bacterial cell wall, and a second or catalytic domain that then degrades that cell wall. If there's sufficient cell wall degradation, the bacterium doesn't have stability anymore and it can die. And so we went to the literature, found a particular endolysin which seemed to have activity against C. perf, and then put our protein engineering tools to work on that. So Seth tested and found that this particular enzyme is indeed active against C. perfringens, but it's only modestly stable. And that's not a good thing for use, but it's also not a good thing for attempts to engineer it to have better selectivity and or potency. So the aim here was to engineer it to be more stable and more active. And so here we again use this combination of combinatorial evolution, but also with a rational design up front. And so Seth grabbed many, many different sequences, a schematic shown here of a handful of them, many different sequences of similar endolysins in nature, and then built a model saying how frequent are particular amino acids at particular sites, but also how frequent are pairs of amino acids co-evolving together. Right? So for example, here it prefers to be isoleucine I, but can change to V. Here it prefers to be glutamine Q, but also can change to V. But importantly, those changes to V happen collectively together. So if we're going to make one of those changes, we should actually make both of those changes. And so this mathematical framework can model that very effectively. And so we put that to use. He also then took some structural information and evaluated where the mutations would be likely to benefit simultaneously with each other. And then went into the lab and experimentally tested these different molecules for how stable they were using a particular yeast assay that I'll skip over now for the sake of time. He then took the functional and the less functional molecules, sequenced them to evaluate how stable they were, and then put that back into this coevolutionary model to make predictions about which mutations we should make and should not make in terms of making a more stable yet still active molecule. And this is what's called the receiver and operating characteristic plot, just showing the prediction quality. And so we want to be basically a step function like that in terms of having a very high true positive rate before we have substantial false positives. And with improved versions of the model, we were able to achieve basically 95% accuracy in that sense. So we then took that model and predicted particular mutations that we could make to stabilize this endolysin antimicrobial, and then tested them. And indeed, long story short, the model worked and the engineering strategy worked in terms of this was the wild type endolysin's activity after being thermally stressed. And this is the activity of several different variants. So the model's not perfect. Some of the molecules do not work. One of the molecules did not work well. One of them only worked moderately better. But the other ones worked dramatically better than our starting point. And so we've improved the stability and activity of this molecule against C. perf. And an analogous example, but with some differences to it, Lena, along with Seth and the group, have engineered additional endolysin, lysi-FM5, against vancomycin-resistant Enterococcus, or VRE, and shown that that is more active than its starting point as well. So this combination of rational guidance, both structurally and genetically, coupled with high throughput evolution, is very powerful to make better antimicrobials. Okay. So lastly, and how am I doing on time? Okay, I'll go very quickly over this one. Can we also do enzyme engineering and make better catalysts? So this is motivated by a particular idea for synthetic urinary reporters, so we can have test the presence of particular biomarkers, not via imaging, but via a particular small molecule showing up in the urine. So the idea would be you administer these engineered molecules to the patient and take a urine test, and the amount of this particular molecule in the urine would be a readout on how much of this biomarker, cancer biomarker, cardiovascular biomarker, what have you, was present, if that works well. Right, but for the sake of time, I'll leave it at that. But what we need for this to work well are very active selective enzymes that can act upon this reporter and generate these small molecule products. And so what Brian, Suvam, and Abby and others have been working on is a way to apply this strategy of rational guided library design with high throughput evolution to enzymes and catalysts, but not antimicrobial enzymes. So in this case, what we want to do is have molecules that degrade this particular covalent bond on this linkage right here. And so what we do is we yeast display our enzyme of interest. In this case, it's called tobacco etch virus, or TEV. But we co-display its substrates. What we want is an enzyme that chews up this particular polypeptide very efficiently and selectively. So the approach here is to co-display both of them, allow proteolysis to happen. If this enzyme is very active, it'll chop this bond right here, and we won't have this little blue tag left anymore. And we can interrogate the presence of that blue tag using binding ligands like antibodies. Instead, if the enzyme is not a very good enzyme, it won't chew anything up, and both of our tags, yellow and blue, will still be present. And so then we can do flow cytometry on these yeast and see which ones have both blue and yellow in high quantity. Those are not good enzymes. And which molecules, or which yeast I should say, have very low levels of blue but very high levels of yellow, i.e. we're effectively proteolyzed at that particular site. I keep stepping on this microphone, I apologize. Right? And so Brian, in this particular case for TEV, did exactly that in the lab using an initially structurally guided approach. So he looked at the structure of the protease, picked particular spots in the vicinity of the substrate. So here the substrate is shown in orange. And so we picked a subset of sites that are adjacent to that active site and mutated them simultaneously, but only this cluster of red and then this cluster of blue, and so on and so forth, and then put it into this evolutionary system, and then tested to see which mutations were more active at cutting the substrate than others. And this is summarized in this very busy heat map, but long story short of this heat map is just simply showing there were mutations relative to the studying enzyme which are much more active than what we had before. And so again, in short, this is working well. We have more to do in terms of now putting these mutations together simultaneously in the same protein and seeing how much more active they are, but so far so good on that front. So with that, in summary, hopefully I've been able to convey some information about protein engineering, in particular to motivate this hybrid or this merger of both rational design approaches and efficient combinatorial approaches, with the summary being we want effective library design, not necessarily effective single variant design. And if we do that, it can empower both improved diagnostics and improved therapeutics in a variety of different areas. So I've tried to highlight some of the people in the lab working on it throughout, a bunch of them shown here at our summer barbecue last summer. I also just want to thank the various funding agencies that support our work, including the department and the U, of course. And I'd be happy to take any questions and also highlight resources you can go to here as well. Thanks for your attention.